So my name's Jay. I work down at the University of Southampton as a clinical research fellow, and as mentioned, I'm wholly funded by the Lewy Bodies Society. So not only am I very grateful for the invite here today to talk, I'm also extremely grateful for the funding for the research project that I'm about to present. I'm going to be talking about inflammation in dementia with Lewy bodies. I think we're around 5 or 10 years behind what we know in Alzheimer's disease in this area. So it really is an exciting area to be involved in. Hopefully, over the next couple of years we'll get more information about what the role of inflammation is in dementia with Lewy bodies, or DLB, and what we could possibly do to target it as a treatment. So I'll just briefly recap a couple of points that Professor McKeith stated, because I'm going to come back to this slide a bit later on. As you all know, alpha-synuclein is the primary pathology in the brains of people with DLB at post-mortem examination. And this type of pathology primarily manifests in Lewy bodies, but there are other types of pathology as well. What I would say is that it's been shown repeatedly that pathology that's more typical of Alzheimer's disease, or AD, in the form of plaques here, made up of amyloid beta, and tangles here, made up of phosphorylated tau. These types of pathologies are actually quite frequently present in DLB, so there is an overlap in terms of pathology between AD and DLB. And I'll come back to this point a bit later. But moving on to inflammation. The immune system is an amazing thing. It works to protect the body from pathogens like bacteria or viruses. It has to do that without actually damaging its own cells, which is quite a tricky balance to play sometimes. And one type of immune cell is the macrophage, and these cells can engulf and destroy things like bacteria or viruses to try and remove them from the body. In the brain, the macrophages are called microglia, and these cells are, there's an example of which is shown here. This is a picture from some brain tissue that's been stained with IBA1, which is a marker of microglia, and you can see the cell body here with some processes coming off that cell. And these microglial cells move around parts of the brain to survey the environment, to look for things that shouldn't really be there, and try to engulf them and destroy them, which is a very helpful thing to do, as you'd imagine. They also do other very helpful things, such as supporting synapses, so the connections between neurons. So they do lots of good things. The worrying thing and the sad thing is that sometimes with age, and also it's been shown in Alzheimer's disease, these microglia can become dysfunctional and lose their neuroprotective properties, and that may lead to neurodegeneration in some of these disorders. I don't really want to labour the points on Alzheimer's disease. I'm at a Lewy body talk, so apologies for having a slide on Alzheimer's disease, but I just want to just paint a brief picture as to where we are at the moment in Alzheimer's disease in this area, so I can go on to talk about where we are in DLB. There have been some large-scale genome-wide association studies, which I'm sure Dr. Brass will talk about a bit later, which have been looking at risk factors for late-onset Alzheimer's disease, and it's been shown that inflammation-related genes have polymorphisms or mutations in some of these genes make you have a higher risk of getting late-onset Alzheimer's disease. That's a really key area of research that's happened over the last few years, which has really pointed us towards inflammation as not only a consequence of Alzheimer's disease, but possibly also contributing to the causation of the disease as well. Some imaging studies have also shown inflammation that may play an important role in Alzheimer's disease, and also some post-mortem brain tissue studies have also shown the same thing. So there is lots of research going on in Alzheimer's disease at the moment, but what do we know in DLB, in dementia with Lewy bodies? Well, the answer is there has been some research. I won't go through this slide in detail, but there have been some post-mortem brain studies in DLB. There's been one imaging study to date, although I know Professor O'Brien is undertaking another one. The sad thing is that a lot of these studies have shown conflicting results. So there's no comprehensive overview at the moment as to what we know in terms of inflammation in DLB. And a lot of the studies are quite small, with numbers less than 12 in each group. So I think it's an important area that we need to establish whether inflammation is important in DLB, and I think we can do that through various different types of studies. And I'm going to be talking about 2 studies that we're doing down in Southampton. So our hypothesis was that inflammation may be different, may be altered in DLB compared to controls, and that the markers that we use to assess inflammation might correlate with the neuropathology of the disease, so the Lewy bodies, the phospho-tau, the amyloid, but also the clinical features, so the symptoms that patients suffer with. I'm examining this through a clinical study and a post-mortem study. The clinical study is still underway, so I haven't got much to say about it. I've just got one slide to give you an overview, a flavour of what we're doing. But hopefully in a year or so I'll be able to present my results. But the post-mortem study is nearing completion, so I can give you some results from that today. So for the clinical study, we're undertaking a cross-sectional observational study, so just looking at one time point and inviting 90 participants to take part. So we have invited 30 people with dementia with Lewy bodies, probable dementia with Lewy bodies, 30 people with probable Alzheimer's disease, and 30 matched controls for a one-off visit where we perform a thorough clinical assessment, and we also do various assessment tools to look at mood, memory, cognition, behaviour, fluctuations using standardised assessment tools. I've listed some acronyms there, apologies for the list, but I'm happy to go through them in more detail if anyone's interested a bit later on. But these are most of them are tools that have been widely used in dementia research. At the same time as doing the clinical assessments, we're also taking blood samples from these patients to look at DNA, which is actually kindly going to be processed by Dr. Brass, who follows me as a speaker this morning, so thank you to him. And also serum and inflammatory markers in terms of immune cells in the blood. So we're going to be looking at how these immune cells react in the laboratory to various stimuli. So we're going to be throwing parts of bacteria at them, parts of viruses, some amyloid and possibly some alpha-synuclein to see whether they react in different ways in Alzheimer's disease and in DLB. And that might hopefully feed into some work developing some biomarkers to aid more accurate and earlier diagnosis. That's a bit of a way off at the moment. I've got some training for those laboratory techniques next week, so as I said, in the next few months, hopefully we'll get some more information in this area. But just moving on to the neuropathology study. So we have obtained post-mortem human brain tissue from 30 post-mortem confirmed DLB cases and 29 matched control cases in one area of the brain, in the middle temporal gyrus. And we have performed immunohistochemistry for a number of markers, so looking at alpha-synuclein for Lewy body pathology, Lewy-related pathology, amyloid beta for the plaques, and phospho-tau for the tangles. So we're looking at the different types of pathology in the brain. We're also looking at markers of inflammation, looking at microglia in particular. So those are the immune cells in the brain that I spoke about. There are 3 markers that I'm using: IBA1, HLA-DR, and CD68. So these 3 markers are widely used in Alzheimer's disease to try and look at these microglial cells in the brain and see what they're doing, whether they're eating up things, whether they're presenting antigens, or whether they're moving around a lot. These are markers that can give us a clue as to what these cells are doing. After we've done the immunostaining, I've taken pictures of the immunostained tissue. So this is an example here of a picture of tissue that's been stained for beta amyloid. There are a couple of plaques that you can see here that are stained brown on some human brain tissue. And we processed all of these images through some computer software to get a black and white image and then a percentage of protein load on the right-hand side. So this gives an indication of how much of that protein is present in that area of the brain. So the higher the protein load, the more beta amyloid or the more tangles. The one marker that we haven't done that process for is for alpha-synuclein for various reasons. But we're using a semi-quantitative assessment. So as suggested in the 2005 consensus criteria, where we are scoring cases from 0 being no Lewy body pathology to 4 being very severe pathology. So I'll just show you what that looks like in terms of results. Across the top row are the markers that we've looked at. So the alpha-synuclein on the left, the amyloid beta in the center, and the phospho-tau on the right. Across the middle are scatter graphs of the control cases on the left and the DLB cases on the right. And at the bottom are some example pictures of some brain tissue that's been stained for those markers. So you can see a few Lewy bodies on the left-hand slide, a nice picture of a plaque in the middle, and some tangles and other pathology on the right-hand side. So what we've found really confirms what I mentioned in the first slide. So I'll just talk through the slide one by one. So in the alpha-synuclein cases, stained cases, we saw no alpha-synuclein staining in the control cases, which is reassuring, and a varying amount in the DLB cases, which is what we were expecting. We saw an increased level of protein load of both amyloid beta and phospho-tau in the DLB group compared to the control group. So that's really as expected. We saw more neuropathology in the DLB group compared to the control group. But when we move on to the microglial markers, we found something a little bit more unexpected. I haven't mentioned that in Alzheimer's disease, these same experiments have been completed and have showed increased levels of HLA-DR and CD68 in Alzheimer's disease, showing that microglia are perhaps phagocytosing more, they are presenting more antigen, they're more active in Alzheimer's disease compared to controls. We didn't find that, so we found no significant difference in protein load in any of the inflammatory markers that we've looked at or that I've presented so far, which is a bit of a surprise, and that starts to... there may be reasons that that's the case, but it also may point us in other directions in terms of inflammation. So lastly, we looked to see whether the neuropathology of DLB is associated with the microglial markers, to see whether there's a link between the two. And again, we did that for one reason, which is that in Alzheimer's disease, it's been shown that if you have more neuropathology, you have more microglial activation in the form of certain markers, including HLA-DR. But we sadly didn't find any association between any of the microglial markers and any of the neuropathology markers. So again, that was quite a surprise. So I'm just going to summarize and speculate as to why I found these findings and what might be causing them. So we've confirmed that there's increased neuropathology in the DLB brain, but that doesn't seem to affect the expression of certain microglial markers that we've looked at. And that may be for a number of reasons, including some limitations of this study. It may also be that we're looking at one time point at the end of DLB at death, at post-mortem, but it may be that inflammation is important in the earlier stages of the disease. And it could be something to do with the areas that we've looked at as well. There may be a possible distinct immunophenotype for DLB that differs from Alzheimer's disease, and that may be confirmed in the clinical study that we're currently undertaking as well. But I'm starting to think that perhaps we need to do a bit more work in terms of other markers of microglia, which we're doing now, but also that there may be other aspects of the pathophysiology of DLB that we need to be focusing on as well, including synaptic alpha-synuclein pathology and possibly the impact of Alzheimer's disease pathology on the Lewy bodies and on cognition. So hopefully with the rest of the work that we're doing in the post-mortem study and also the clinical study that we're doing, we'll be able to give you a much more comprehensive picture of the role of inflammation in DLB in the next year or so. So thank you very much. I'm just going to say thank you to a few people just briefly. So thank you to my supervisors, Professor Holmes and Dr. Delphine Bosch, our collaborators, the brain banks that have provided brain tissue, and also a huge thank you to the Lewy Bodies Society for funding the research that I'm undertaking. And thank you all for listening as well.